Welcome to the deep dive. You're probably here because, like us, you're right on the cutting edge of longevity research. And this week, we've sifted through the latest publications to bring you the findings that might actually change how we think.
Yeah, our mission is pretty straightforward: to efficiently break down the most potentially impactful new research for, people working in longevity biotech.
We've looked at the papers listed on longevitypapers.com, focusing on just the last few days, basically Monday, March 31st, up to today, April 3rd, 2025.
That's right. Pulled from PubMed and also bioRxiv, just to be clear, this BIO archive. We're really looking for credible work from solid institutions, but also work that's maybe a bit surprising, something that might hint at a necessary shift in our current models.
Okay, but let's manage expectations here. Honestly, truly game-changing papers that rewrite the textbooks overnight, those are super rare. Finding one in a single week's publications is, well, unlikely.
Definitely. What we usually see, and what we've mostly seen this week, is incremental progress. Important steps, new data points, but steps nonetheless.
Exactly. So our aim today is to critically evaluate the most interesting stuff we found, look at the potential, sure, but also really scrutinize the limitations.
So with that reality check in place, shall we jump in?
Let's do it. What's first?
Okay. The first one that stood out is titled Inhibition of Ferroptosis Delays Aging and Extends Health Span Across Multiple Species.
Ferroptosis, okay?
Yeah. The authors are Haijun Fu, Xingyu Zhu, Dilin Qin, and quite a few others. They're based at Southwest Medical University in Luzhu, Sichuan, China. And this was published just this Monday in Advanced Science.
Right. Ferroptosis is that iron-dependent type of cell death. We know it's regulated. So what's the big finding here?
Well, the core idea they're putting forward is that ferroptosis isn't just associated with aging, but it's actually a key driver of cellular senescence.
Senescence.
Yeah.
So cells stop dividing, hang around, cause inflammation,
Right.
that whole cascade. Linking ferroptosis directly as a driver is significant.
It is. And what makes this study particularly compelling, I think, is the breadth of their evidence. They used inhibitors of ferroptosis compounds called leproxstatin-1 and ferrostatin-1.
Okay.
And they show these inhibitors work to delay senescence and improve health span markers, not just in one system, but across several. They tested them in cultured human fibroblast cells.
In vitro, standard stuff.
Right. Then in vivo in C. elegans, the nematode worms,
Classic aging model.
and also in various mouse models. So this multi-level validation really strengthens their case. They're basically showing this iron-dependent lipid peroxidation, which damages cell membranes, seems tightly linked to cellular aging itself.
That cross-species validation is definitely a plus. Did they see effects beyond just lifespan in the worms? Sometimes lifespan extension comes at a cost.
Good question. Yes, they did. In C. elegans, the treated worms didn't just live longer, they also showed improved locomotor activity. They moved better.
Okay, that's good.
They boosted reproductive output, and importantly, less accumulation of lepofusin.
The age pigment.
Right. So signs of generally better health, not just dragging out old age.
Exactly. And in the mouse models, at least over the 6 months they studied them, things also looked promising. No obvious negative effects on body weight or liver and kidney function.
6 months is decent for a mouse study, but not exactly long-term for translating to humans.
True. But interestingly, they even saw improvements in some hematological parameters like blood cell counts.
Oh, that could hint at effects on maybe hematopoietic stem cells, since those decline with age.
It's possible. That certainly warrants a closer look whether ferroptosis is involved in stem cell aging. But the big picture here for longevity research is pretty exciting.
You mean if ferroptosis is a fundamental mechanism and we have drugs that hit it effectively across species?
Then it becomes a really attractive therapeutic target, right? Getting at a root cause, potentially, instead of just managing symptoms.
It fits nicely with the broader focus on cellular senescence, too. We know senescent cells cause trouble if ferroptosis helps create them. Then inhibiting ferroptosis could prevent that trouble at the source. It could have really wide-ranging benefits against age-related diseases.
Okay, this sounds almost too good. Time for the skeptical hat. What are the potential pitfalls or unanswered questions? You mentioned the 6 months in mice.
Right. 6 months isn't forever. We absolutely need longer-term studies, especially before thinking about humans. What are the consequences of chronically blocking a cell death pathway?
Could it interfere with necessary cell turnover? Or maybe increase cancer risk down the line if damaged cells aren't cleared?
Exactly. And how specific are these inhibitors, really? Like leproxstatin-1, ferrostatin-1? Could they be having off-target effects we don't know about yet? That's always a concern.
And is this idea completely novel, targeting cell death and aging?
Well, no, not entirely. Apoptosis, another form of programmed cell death, has been studied in aging for a while.
Mm.
But the specific focus on ferroptosis and the consistency they're seeing across different models, human cells, worms, mice, that does feel like it could be a significant new angle.
So if we were designing the next steps, the follow-up studies, what would be top priority?
Several things. Definitely longer-term studies in larger animals, rats, maybe even non-human primates, to get a better handle on efficacy and, crucially, long-term safety.
Makes sense. Closer to human physiology.
Also, combination therapies. Could we combine a ferroptosis inhibitor to prevent senescence with a senolytic drug to clear existing senescent cells?
Like a 1-2 punch.
Potentially. We also need to understand the mechanisms better. Can we find reliable biomarkers for ferroptosis activity in aging tissues? That would help track the effectiveness of treatments.
And dig deeper into the signaling pathways,
Mm.
like how ferrostatin-1 boosts GPX4, that protective enzyme.
Precisely. Understanding that pathway might reveal other druggable targets.
Mhm.
And we need to look at interactions with other pathways too, apoptosis, autophagy, do they work together against each other?
Lots to unpack there, and more detailed long-term health span measures too, right? Not just lifespan.
Absolutely. Cognitive function, physical performance, reproductive health, track everything over the entire lifespan in worms and mice. Does it really mean more healthy years?
That's always the crucial question. So for our listeners, this paper's strength seems to be its multi-level approach: cells, worms, mice. How convincing is it, though, that ferroptosis is truly driving senescence, not just correlated with it? It's certainly food for thought.
Definitely a major point to consider.
Okay, let's switch gears. Paper number 2, also from Monday, March 31st.
Correct. This one's titled, Doxyfluoridine Promotes Host Longevity Through Bacterial Metabolism. It's from Rui Wei, Yuling Peng, Yami Luo, and colleagues at West China Hospital Sichuan University in Chengdu, published in PLS Genetics.
Doxyfluoridine, bacterial metabolism.
Oh, good. Yes.
Longevity? That sounds like an unusual combination.
It really is. So doxyfluoridine is a fluoropyrimidine derivative. The researchers found it rescues age-related alternative splicing defects in C. elegans.
Okay, splicing defects when gene messages get jumbled with age. We know that's a hallmark. And rescuing that extends lifespan.
Yes, it extends their lifespan. But here's the truly surprising part. The drug's ability to do this seems entirely dependent on the bacteria living in the worm's gut.
Wait, what? The gut microbes have to process the drug for it to work on the worm's aging?
That's what the study strongly suggests.
Mm.
Bacterial metabolism is apparently essential. It throws open this really fascinating possibility. Could we target the host microbe interaction itself as a longevity strategy?
Wow, that adds a whole layer of complexity, but
Oh.
immediate skepticism kicks in. Human microbiomes are wildly diverse. How likely is it that what works via specific bacteria in a worm translates to us?
That's the million-dollar question, isn't it?
Yeah.
Translatability is a huge hurdle here. Different bacteria might metabolize the drug differently or maybe produce unwanted, even toxic byproducts in humans.
Yeah.
But on the other hand, it fits with the growing realization of just how important the gut microbiome is for overall health and aging. We know it influences immunity, metabolism, even brain function. So the concept isn't totally out of left field.
Is this specific mechanism a longevity drug needing bacterial processing? Is that novel?
As far as I know, for a longevity effect, this specific dependence on bacterial metabolism seems pretty new. We know gut bacteria can affect how other drugs work, but making it essential for lifespan extension is a novel twist.
You mentioned some specific, intriguing details from the paper.
Yeah, a few things really stood out. First, if they use worms that lacked live bacteria, the doxyfluoridine had no effect on
But
lifespan.
none at all. So the bacteria have to be alive and active.
Exactly, which makes you wonder, how are live bacteria processing it differently? Is it purely enzymatic, or is there active signaling involved? That needs more work.
What else?
They also pinpointed specific bacterial genes, names like IJJG-UP and UDP, involved in nucleotide salvage pathways. If these genes were mutated in the bacteria,
The drug didn't work as well.
right, its lifespan extending effect was reduced. This suggests the bacteria need these specific pathways to convert doxyfluoridine probably into its active form, FUMP, or something related.
Okay, so specific bacterial biochemistry is required. Any other clues?
Yes, they found that giving the worms extra uridine completely blocked the lifespan extension from doxyfluoridine.
Uridine. Suggesting competition? Maybe? Like the drug's metabolites interfere with how the worm uses uridine?
That's a strong possibility. It also implies that diet, or nutrient availability could potentially influence how well a drug like this works.
Interesting. And did they identify what the bacteria were producing from the doxyfluoridane?
They found increased levels of a couple of bacterial metabolites after treatment, agmatine and linoleic acid.
Agmatine, I think I've heard that mentioned in C. elegans lifespan studies before, linked to neurotransmitters.
That's right. It's known to extend lifespan in worms. Linoleic acid's role is maybe less clear. A key question is, are these metabolites absorbed by the worm directly, acting on splicing, or do they trigger some signaling cascade between the bacteria and the host?
So are the bacteria like little drug factories inside the worm?
That's a great way to put it. They also did proteomics on the bacteria and found changes upregulation of nitrogen metabolism and the TCA cycle. How those bacterial shifts benefit the host isn't totally clear yet. Maybe changing nutrient supply. It's complex.
Incredibly intricate. Does doxyfluoridane have any clinical background? Is it used in humans?
It does. It's apparently less toxic than a related drug, FENDR, and it has been tested in phase 2 and 3 trials for other indications, though not longevity.
So there's some existing data.
Yes, which might help if anyone pursued this further. But that microbiome dependence is a massive challenge for personalized medicine in aging. How do you ensure the drug works consistently when everyone's gut bacteria are different?
A huge hurdle. But the paper did highlight alternative splicing as a potential universal aging issue, right? And they had a neat tool.
Yes, the idea that splicing errors accumulate with age across species is gaining traction. And they developed a clever dual fluorescent reporter system in the worms to screen for drugs that fix these errors. That tool itself could be valuable for finding other longevity compounds.
So even with the translation question marks, this paper opens a really novel door, targeting that host microbe axis for aging.
Definitely. It makes you wonder what other biological partnerships we haven't fully appreciated might be key players in how we age.
Okay, so those were the two main papers we wanted to deep dive on. But just for balance, were there other interesting, maybe more incremental findings this week?
Yeah, a few others are worth a brief mention. One in autophagy looked at how signals from bone, specifically the hormone osteocalcin, are sensed by neurons in the hippocampus via their primary cilia, boosting autophagy and potentially protecting cognition.
Bone-brain connection via autophagy. Interesting link, but maybe not a direct overall lifespan paper yet.
Right. Then another in aging cell focused on senescent endothelial cells in the brain's blood vessels. They showed these are key drivers of blood-brain barrier breakdown and cognitive issues in aged mice. Using senolytics to clear them improved neurovascular function.
Promising for brain aging, but we know senolytics can have variable effects in different tissues, don't they?
Exactly. Results aren't always consistent body-wide. And finally, a paper in aging did a really deep dive using single-cell analysis on mouse T cells across different ages.
Immune aging, always important.
Yeah, they mapped out detailed changes linked to immune decline, potentially identifying pathways for rejuvenation therapies. But again, translating detailed mouse immune findings to the complexity of the human immune system is always a big step.
Okay, so putting it all together, it sounds like our initial prediction held true. Interesting findings, definite progress, but nothing this week that screams rewrite the models?
I think that's fair. The ferroptosis paper and the doxyfluoridine microbiome paper are certainly thought-provoking, presenting potentially novel strategies. But they both need a lot more research, replication, and long-term validation.
We have to keep that critical perspective. Promising initial results often face hurdles down the line, especially when translating to humans. The probability of any single finding dramatically extending human lifespan remains, realistically, quite low.
Absolutely. Maintain healthy skepticism, appreciate the incremental gains, and understand the uncertainties.
So, key takeaways. Ferroptosis inhibition looks interesting as a potential cross-species target, worth watching closely. And the doxyfluoridine study throws a fascinating spotlight on the gut microbiome's role, even if translation is tricky.
And it all underscores how science progresses bit by bit, study by study, refining our understanding of this incredibly complex thing called aging.
Right. So maybe a final thought to leave our listeners with, building on that doxyfluoridine paper. If the host microbe link is that critical, what other unexpected biological partnerships, maybe involving other symbiotic organisms or even interactions between different organ systems we haven't fully explored, might hold keys to longevity? It pushes us to think more ecologically about aging, doesn't it?
It really does. A much broader context to consider.
Well, that's all the time we have for this deep dive. Thanks for tuning in.